Hey science fans! Welcome back to another exciting journey into the world of science. Today, we're diving into the incredible world of DNA replication, a process that is fundamental to all living organisms. DNA holds the instructions for life itself. It's like the ultimate guidebook that tells every cell in your body what to do. It's the blueprint for everything that makes you, you. From the color of your eyes to the way your heart beats, it's all encoded in your DNA. DNA replication is like a high-tech photocopier that duplicates the instruction manual so that each new cell has its own copy. Without DNA replication, cells couldn't divide, and life as we know it wouldn't exist. Pretty neat, right? So next time you think about the complexity of life, remember the unsung hero, DNA replication. Now, let's get down to the nitty-gritty of how this copying process actually works. DNA replication is a fundamental process that occurs in all living organisms, ensuring that genetic information is accurately passed from one generation to the next. Imagine DNA as a twisted ladder. This iconic double helix structure is composed of two long strands that wind around each other, forming a stable and compact shape. This ladder has two long strands, and each strand is made up of building blocks called nucleotides. These nucleotides are the essential units of DNA, consisting of a sugar, a phosphate group, and a nitrogenous base. During DNA replication, this twisted ladder unzips, separating the two strands. Enzymes break the hydrogen bonds between the base pairs, creating two single strands that serve as templates. Each strand then acts as a template for building a brand new matching strand. Enzymes called DNA polymerases add complementary nucleotides to each template strand. It's like having two half ladders, and we use each half to build a whole new ladder. This semi-conservative method of replication ensures that each new DNA molecule contains one original strand and one newly synthesized strand. This process ensures that each new DNA molecule is an exact copy of the original. The accuracy of DNA replication is crucial for maintaining genetic stability and preventing mutations. We start with one DNA molecule and end up with two identical DNA molecules. This doubling of the genetic material is essential for cell division, allowing organisms to grow, develop, and reproduce. Now meet the enzyme superstar, DNA Helicase. This molecular machine is responsible for unzipping the DNA double helix, like a zipper slider cruising down a jacket. It breaks the bonds holding the two strands together, creating a replication fork. Imagine DNA Helicase as a tiny, determined construction worker with a crowbar. It pries open the double helix, exposing the individual nucleotide bases. Without DNA helicase, the replication process would come to a screeching halt. This unzipping action is crucial because it allows other enzymes to access the single strands and start building the new DNA strands. Way to go, DNA helicase! Next up, we have DNA polymerase, the master builder of the replication process. This enzyme is responsible for assembling the new DNA strands by adding nucleotides one by one. Think of DNA polymerase as a highly skilled bricklayer. It carefully reads the original DNA strand and then adds the matching nucleotide to the new strand. It's like a game of molecular Tetris, ensuring that each new block fits perfectly. DNA polymerase is incredibly accurate, making very few mistakes. It also has a proofreading function, double-checking its work to ensure the new DNA strand is an exact copy of the original. Talk about attention to detail. Section 5. Holding it together, DNA clamp and SSB proteins. As the DNA strands are being replicated, we need some helpers to keep things stable. That's where DNA clamp and single-stranded binding proteins, or SSB proteins, come in. Imagine DNA clamp as a tiny clothespin, clamping onto DNA polymerase and preventing it from falling off the DNA strand. It keeps DNA polymerase securely attached, allowing it to do its job efficiently. Meanwhile, SSB proteins act like little pieces of tape, binding to the separated DNA strands and preventing them from coming back together. They keep the strands separated, providing a clear template for DNA polymerase to work its magic. Section 6. Relieving the twist topoisomerase. Now, imagine trying to unwind a long, twisted rope. You'll eventually encounter some serious tension. The same thing happens with DNA as it unwinds during replication. That's where topoisomerase comes to the rescue. Think of topoisomerase as a molecular stress ball. It cuts and rejoins the DNA strands, relieving the tension that builds up during unwinding. This prevents the DNA from getting tangled up and ensures that replication can proceed smoothly. Without topoisomerase, the DNA molecule could break under the strain, putting a stop to the whole replication process. It's the ultimate tension tamer. Section 7. Setting the stage primase. Before DNA polymerase can start building the new DNA strand, it needs a little something to get it going. It's like needing a primer before you paint a wall. That's where primase comes in. Think of primase as a tiny painter's tape, laying down a short RNA primer on the DNA strand. This primer provides a starting point for DNA polymerase to attach to and begin its work. Without primase, DNA polymerase wouldn't know where to start building the new DNA strand. It's the essential starting block for DNA replication. Section 8. Sealing the deal, DNA Ligase. As the new DNA strands are being built, there are tiny gaps that need to be sealed up. That's where DNA Ligase swoops in to save the day. Think of DNA Ligase as a tiny molecular glue stick. It seals the gaps between the DNA fragments, creating a continuous and complete DNA strand. It's the final step in the DNA replication process. Without DNA Ligase, the new DNA strands would be fragmented and incomplete. It ensures that the newly replicated DNA is strong and stable. Section 9. Passing on the blueprint. The importance of DNA replication. So there you have it, DNA replication in all its glory. This process is fundamental to all living organisms, from the simplest bacteria to the most complex plants and animals. It ensures that genetic information is accurately passed from one generation to the next. From unzipping to building to sealing, it's a complex and fascinating process. The DNA double helix unwinds, and each strand serves as a template for a new complementary strand. Enzymes like DNA polymerase add nucleotides to form the new strand, while DNA ligase seals any gaps. The significance of DNA replication cannot be overstated. It is the foundation of biological inheritance, allowing organisms to grow, develop, and reproduce. Without this process, life as we know it would not be possible. DNA replication ensures that every new cell gets a complete set of genetic instructions. This is essential for the proper functioning of cells and the overall health of an organism. Each time a cell divides, it must replicate its DNA so that the two daughter cells receive identical genetic material. This is crucial for growth, repair, and the continuation of life itself. For instance, when a plant grows from a seed, its cells are constantly dividing and replicating their DNA. Similarly, in humans, DNA replication is vital for processes such as wound healing and the development of a newborn baby. Without it, cells wouldn't be able to divide, and organisms wouldn't be able to grow or reproduce. The continuity of life depends on the accurate and efficient replication of DNA. Any errors in this process can lead to mutations, which may cause diseases or developmental issues. From the tiniest bacteria to the largest whale, DNA replication is the unsung hero of life. It operates behind the scenes, tirelessly working to ensure that every living organism can thrive and adapt to its environment. This process is a testament to the incredible complexity and precision of biological systems. It's a process that happens countless times every second, ensuring that the blueprint of life is passed on to future generations. Pretty amazing, isn't it? The next time you see a plant growing, an animal maturing, or even a simple wound healing, remember the vital role that DNA replication plays in making it all possible. Truly, it is one of the most remarkable processes in the natural world.